The University of Utah has received its first ever FDA 510(k) clearance for the bone bolt system, a technology developed by its orthopedics department and Orthopedic Innovation Center. The bone bolt system utilizes titanium alloy implants and surgical tools to hold fractured bones in place, aiding in the healing process. It is designed for difficult-to-treat breaks such as those in the pelvis and long bones of arms and legs. The FDA clearance allows its use for repairing fractures in various parts of the skeleton, excluding unfused growth plates, and can be used in patients as young as 2 years old. With the approval, the university will work on commercializing the bone bolt system by partnering with the orthopedics industry. A new article highlights the difficulties in treating sarcoma, a rare and diverse type of cancer. Recent breakthroughs such as targeting the lingo-1 protein for Ewing sarcoma and developing new drugs like TP1287 and NY-ESO1 TCRI-L15NK show promising steps toward better treatment options in the future. Researchers from the Keck School of Medicine have discovered that cell stress can activate a protein called GRP78, causing it to migrate to the cell nucleus and alter genes. This gives the cell metastatic properties, promoting cancer spread. The finding opens up possibilities for preventing metastasis by targeting GRP78 and its interactions with other proteins in the nucleus. This research could lead to the new ways of tackling cancer. Researchers from New Zealand's Ferrier Research Institute, the Millikan Institute of Medical Research, and Australia's Peter Doherty Institute for Infection and Immunity have developed a novel mRNA vaccine for malaria, drawing inspiration from cancer immunotherapy techniques. The vaccine showed promising results in mouse models, generating an immune response and preventing infection with Plasmodium bergei, a form of the malaria parasite. Notably, the vaccine remained effective even in animals previously exposed to malaria, setting it apart from other vaccines. Unlike the current malaria vaccine Mosquirix, which has limited efficacy, this mRNA vaccine encodes the entire malaria protein and aims to upregulate memory T cells in the liver, providing better immunity. The researchers plan to conduct humanized mouse model tests and potentially enter clinical trials in the coming years. German researchers from the Karlsruhe Institute of Technology have investigated the impact of biochar, a phosphorus-based alternative on plant growth. Biochar, a product of biomass pyrolysis, has been studied as a replacement for phosphate fertilizers. The researchers found that the origin of the biochar biomass is crucial for its symbiosis with naturally occurring arbuscular mycorrhizal fungi, or AM fungi, in the soil. In experiments with tomato seedlings, biochar made from chicken manure, rich in phosphates, hindered the symbiosis with AM fungi, while biochar from wheat straw enhanced the symbiosis. Understanding this complex molecular response may eventually lead to programming plants to require less phosphate, reducing mineral fertilizer use. The research is part of the Karlsruhe Institute Future Fields project, Plants Fit for the Future, aiming to prepare plants for the future with limited phosphorus fertilizers and reduced pesticide use. Merck and Moderna have launched a phase 3 trial for the personalized cancer vaccine V940 in combination with Keytruda for high-risk melanoma patients. The vaccine, which showed promising results in phase 2b, will now include a stage 2b for patients in the study. The trial aims to enroll 1,089 patients across multiple countries with the primary goal of recurrence-free survival. Secondary objectives include distant metastasis-free survival and overall survival. The FDA and the European Medicines Agency have granted priority designations for the vaccine combo. The study is set to continue until October 2029, and plans are in place to explore its potential in other cancer types, including non-small cell lung cancer. Novartis and Danish health-focused software developer Down Health have launched a new app called Ekiva MS, designed to help people with multiple sclerosis better understand their disease and improve their care. The app collects health and behavioral data from AMOS patients, allowing them to track changes in symptoms and quality of life, while also using smartphone sensors to capture additional day-to-day information. It integrates with other health tracking tools, including Dreams by Helios AG, which measures movements, balance, dexterity, and vision as digital biomarkers. As Ekiba MS gathers more information, it offers symptom management tips and compiles data into a report to be shared with healthcare providers for more personalized treatment plans. The app is initially available for free download in Germany. Novartis has been actively engaging the digital health space, collaborating with various tech developers for digital therapeutics and health management platforms. Tarsus Pharmaceuticals has received FDA approval for XDEMVY, the first treatment for Demodex blepharitis, an eyelid disease that affects around 25 million Americans. The eye drops directly target Demodex mites, the cause of the disease, that are expected to be available by the end of August. The company has launched marketing campaigns to raise awareness about the condition, which is often misdiagnosed. Clinical trials involving over 833 patients showed improvements in collarettes, the accumulation of mite waste and eggs. Previously, patients relied on tea tree oil and lid wipes, which did not address the root cause of the disease. Researchers from Switzerland have developed a new 3D printed material for sensors and displays that not only changes color based on temperature and tensile stress, but also conducts electricity and is biodegradable. The material is created from a combination of cellulose and carbon nanotubes, resulting in liquid crystals that exhibit structural colors similar to the coloring seen in peacock feathers and butterfly wings. By adding carbon nanotubes, the liquid becomes electrically conductive, allowing the researchers to control the temperature and color of the liquid crystals through the application of an electric voltage. The addition of cellulose nanofibers makes the mixture 3D printable without affecting its color or conductivity. Potential applications for the cellulose-based ink include temperature and deformation sensors, quality control in food, and biomedical diagnosis. The researchers plan to explore further possibilities and phenomena related to structural color and its applications in sustainable and biodegradable electronics and the Internet of Things. Researchers from the University of Duisburg-Essen, ETH Zurich, and Ruhr University Bochum have developed a new way to detect viruses and bacteria using carbon nanotubes modified with DNA anchors. The nanotubes can be customized to react specifically to certain pathogen molecules and emit a fluorescent signal in response. By attaching recognition units such as antibodies to the DNA anchors, the nanotubes can interact with specific bacterial or viral molecules, altering their fluorescence. The researchers utilize DNA structures with guanine quantum defects to enable easy customization of the carbon sensors for different target molecules. The new sensor principle was successfully demonstrated with the SARS-CoV-2 protein using aptamers as recognition units. The sensors showed reliable detection of the SARS-CoV-2 protein and exhibited higher selectivity and stability in solution compared to sensors without guanine quantum defects, making them suitable for complex environments such as blood and organisms. A study presented at Nutrition revealed that adults with mild cognitive impairment who received the probiotic Lactobacillus rhamnosus GG for 3 months showed improvements in cognitive scores compared to those who received a placebo. The improvement in cognition was linked to changes in the participants' gut microbiome. Prevotella, a type of gut bacteria, was found to be more present in participants with mild cognitive impairment. Those who received the LGG probiotic experienced a decrease in Prevotella levels and an increase in cognitive scores. The findings suggest that the gut microbiome may play a significant role in early cognitive impairment, and interventions like probiotics or diet changes could potentially slow down cognitive decline. Further research is needed to confirm these results and implement microbiome-focused interventions for cognitive health management. The Ice Cube Neutrino Observatory in the Antarctic has created the first ever image of the Milky Way using neutrinos. Neutrinos are high-energy particles that provide information about extremely energetic processes in the universe. The detector comprises over 5,000 light-sensitive sensors, captures neutrinos arriving from space to Earth. The team developed, analyzes, and utilized machine learning to extract the faint signal of the Milky Way from the background noise and to improve the identification of neutrinos. These findings offer insights into cosmic radiation and could advance the exploration of our cosmic neighborhood. Researchers at the Karlsruhe Institute of Technology have developed a new class of biomaterials using enzyme foams which exhibit exceptional durability and activity. These materials offer promising applications in industrial biotechnology, material sciences, and food technology. The enzymes in the foams maintain their catalytic activity and stability thanks to a unique protein network formed during the foaming process. The discovery opens up new possibilities for efficient enzyme use in large-scale applications and simplifies production and shipping processes. A breakthrough in Alzheimer's treatment, the antibody-based drug donanemab has been shown to slow the progression of early-stage dementia by 35% in a clinical trial. Experts from the German Society of Neurology have evaluated the study's results. Donanemab targets beta-amyloid, a protein associated with Alzheimer's plaques in the brain. The trial demonstrated significant benefits in slowing cognitive decline and reducing amyloid plaques. While the treatment shows promise, there are challenges to address, such as high therapy costs and potential side effects, including cerebral microhemorrhages. The researchers emphasized the importance of prevention efforts, as a significant percentage of dementia cases could be avoided through lifestyle factors like a balanced diet and regular mental and physical activities. Scientists from Children's Hospital of Philadelphia and the University of Pennsylvania Perelman School of Medicine have developed a promising proof-of-concept mRNA-based in vivo gene therapy for sickle cell disease. The therapy uses mRNA encapsulated in a lipid nanoparticle targeted to hematopoietic stem cells to correct the gene mutation responsible for sickle cell disease. In tests on human HSCs, the therapy showed around 90% effectiveness. This in vivo gene therapy offers advantages over current bone marrow transplants and experimental gene therapies, as it avoids the need for chemotherapy and prolonged hospital stays. The researchers aim to conduct further animal studies, including non-human primates, to determine its potential in humans. If successful, this approach could provide a more affordable and efficient treatment for sickle cell disease compared to existing gene therapies. Researchers from ETH Zurich have developed a battery-powered device called Direct Current or DC Actuated Regulation Technology, or DART, that stimulates gene expression and prompts cells to produce insulin in diabetic mice. The device uses an electric current to stimulate bioengineered cells, allowing for fine-tuning of gene expression. The team used ROS, or reactive oxygen species, which are produced naturally in the body, to trigger gene expression in insulin-secreting cells. In tests on mice with type 1 diabetes, the device was able to control blood sugar levels effectively. The technology has several advantages, including low power consumption and the ability to provide therapeutic insulin injections for up to 5 years using a small battery pack. The researchers believe this technology could pave the way for wearable electronics to directly program cell and gene therapies in the future. Quest Diagnostics has introduced the AD-Detect test, a direct-to-consumer blood test that assesses a person's Alzheimer's disease risk by measuring beta-amyloid protein levels. The test calculates the beta-amyloid 40-to-48 ratio, indicating the risk of developing Alzheimer's. Although not a diagnostic test, it can help individuals understand their risk. The test costs $399 and is recommended for those with a family history of Alzheimer's, aged 65 and above, or with a history of brain trauma. This launch comes amid increased interest in Alzheimer's treatments, with recent FDA approval for therapeutic antibodies targeting beta-amyloid plaques. Thanks for watching my Biotech News Update. If you found this video informative, please don't forget to hit the like and subscribe button for more updates. Additionally, all the sources used in this video can be found in the description below. Stay tuned for more exciting news in biotech.